Welcome student. Today we discuss about DNA packaging in eukaryote. DNA packaging in eukaryote, but before that, eukaryote means that cell which is the well organized cell or well developed cell here, and in that the nucleus is well developed. That is the well organized nucleus, and in that nucleus the nuclear membrane is present, nucleolus is present, and the thread like structure, that is the chromatin material, is present in the form of chromosome. अशा प्रकारचं जे न्यूक्लियस आहे की ज्याचं न्यूक्लियर मेम्ब्रेन त्याला आहे, पटल त्याला आहे, त्याच्यामध्ये न्यूक्लिओलस आहे आणि क्रोमॅटिन मटेरियल आहे, दॅट इज द थ्रेड लाईक क्रोमोसोम्स आहेत. अशा प्रकारचं न्यूक्लियस, दॅट इज द वेल ऑर्गनाइज्ड न्यूक्लियस, हे युकॅरियोटिक सेलमध्ये असतं. अँड इन युकॅरियोट देर इज DNA विच इज मच मोर कॉम्प्लेक्स. युकॅरियोटचा DNA जो आहे तो मच मोर कॉम्प्लेक्स आहे, and that DNA is associated with histone and non-histone protein. DNA that which is associated with, that is the histone and non-histone protein, and it is reported by the Kornberg in 1947. Then what is histone and non-histone protein? But here we discuss that is the histone protein here. And what is histone? That histones are the protein. Histones are the protein, and which is the charge which acquired its charge. And what charge it's acquired? It acquired positive charge, because DNA is the negatively charged, or DNA is the negative charge, and DNA which is the negative charge, negative and positive get attached or get bind together. Why? Because negative not bind together, or positive not bind. That is the negative and positive. That's why DNA is the negative charge, and that binding proteins which is the positive charge, and that proteins which is made up of lysine and arginine. प्रोटीन हे कशाने बनलेले आहेत? लायसिन आणि आर्जिनाइनने बनलेले आहेत, and both are the basic amino acids. That is the basic amino acids आहेत lysine and arginine, and this histone which which is the 8 molecules known as histone octamer. हे हिस्टोन जे आहे ते 8 molecules नी बनलेले आहे, that is the histone octamer. Nucleosome, what is the nucleosome? Which is the negatively charged DNA which is wrapped around the positively charged histone octamer, forming a structure known as nucleosome. आपण त्याची डायग्राम पुढे बघणार आहोत. परंतु न्यूक्लिओसोम हे काय आहे? जशी महादेवाची पिंड असते, पिंड, आणि त्याला सापाने जसा वेटोळा साप जसा वेटोळा घालतो तशा प्रकारचं स्ट्रक्चर, दॅट इज द न्यूक्लिओसोम. म्हणजेच DNA हे काय होतात? न्यूक्लिओसोम जे आहे हे आत हिस्टोन आहेत, प्रोटीन आहेत, 8 ऑक्टॅमर्स आहेत. 8 जे आहेत ते हिस्टोन प्रोटीन आत आहेत, आणि त्याच्या कडनी हे काय झालेलं आहे? DNA रॅप होत आहे किंवा कडनी गुंडाळलं जात आहे DNA. त्या एकंदरीत एका स्ट्रक्चरला न्यूक्लिओसोम असं म्हणतात. देर आर मेनी न्यूक्लिओसोम्स बट वन, दॅट इज द 8 ऑक्टॅमर हिस्टोन and wrapped DNA. That structure is called a nucleosome. That is the compaction of positively charged histone protein and negatively charged DNA, which is called nucleosome. Here DNA is the negative charged molecule. Because why DNA is negative charge? Because DNA is, that is the negative charge because of phosphate group. Phosphate group असल्यामुळे, that is the PO4-3, that phosphate group, because of that phosphate group DNA is the negative charge. That protein which is the positive charge, protein is lysine and arginine rich basic protein, which is the called histone protein. Here DNA packaging in eukaryote, that is the histone protein. We know that the histone protein, there are 5 types of histone protein. in the packaging of DNA. There are 5 types of histone protein in the packaging of DNA, that is the H1, H2A, H2B, H3, H4. These 5 proteins which are in the packaging of DNA here, and 4 of them, ह्याच्यातले 4 जे आहेत, that is the H2A, H2B, H3 and H4 occurs in a pair. हे पेयर्स मध्ये असतात, म्हणजे जोड्या. that is the H2A 2 proteins, H2B 2 proteins, H3 2 proteins and H4 2 proteins which to produce histone octamer. आपण म्हणत होतो की DNA हे रॅप होतं हिस्टोन ऑक्टॅमरला, तर या हिस्टोन ऑक्टॅमरला DNA काय होते, गुंडाळलं जाते. म्हणजेच H2A चे 2 प्रोटीन, H2B चे 2 प्रोटीन, H3 2 प्रोटीन, H4 2 प्रोटीन, ऑक्टॅमर फॉर्मेशन होते. हे 4 आणि त्याच्या दुप्पट, दॅट इज द 8, दॅट इज द टोटल कॉल्ड, म्हणजेच त्याला काय म्हणतात, दॅट इज द कोर ऑफ न्यूक्लियोसोम. त्यालाच कोर ऑफ न्यूक्लियोसोम असं म्हणतात. अँड अबाउट 200 base pair of DNA is wrapped over one nucleosome. एक न्यूक्लियोसोम जे आहे, ऑक्टॅमर जो आहे, हिस्टोन प्रोटीन जो आहे, त्याला DNA जे गुंडाळलं जाते, रॅप होते, त्या DNA मध्ये फक्त त्या रॅपिंग झालेल्या DNA मध्ये 200 बेस पेअर असतात. दॅट इज द 200 बेस पेअर अँड विच इज 1/3 दॅट इज 1.75 टर्न्स. एका DNAचा जो रॅपिंग झालेला आहे तो हाऊ मेनी टर्न, दॅट इज 1.75 टर्न एवढा DNA रॅप झालेला असतो. आणि 1.75 टर्न DNA मध्ये 200 बेस पेअर असतात. दॅट इज द 2 ॲडजस्टंट न्यूक्लियोसोम इज कॉल्ड लिंकर DNA. जशा प्रकारची मण्यांची माळ असते, 2 मणी आणि त्याच्यामधला जो दोरा असतो, त्या म्हणजे एक न्यूक्लियोसोम, त्याच्यानंतर दुसरा न्यूक्लियोसोम, त्याच्यानंतर तिसरा न्यूक्लियोसोम, अशा प्रकारची सुंदर अशी मण्यांची माळ, तशा प्रकारचं काय असते, DNA पॅकेजिंग असतं. त्याच्या 2 न्यूक्लियोसोम मधला जो DNA आहे, दॅट DNA विच इज कॉल्ड लिंकर DNA. दॅट इज द 2 ॲडजस्टंट न्यूक्लियोसोम इज कॉल्ड लिंकर DNA. अँड हिअर H1 histone protein, we talk about histone protein which is the 5 histone protein, and in octamer that is the only 4 that is the occurs in a pair, that is the 4 protein. But here H1 histone protein which called plugging protein and acts as a marker protein. म्हणजे ह्या न्यूक्लियोसोमच्या कडला जो हिस्टोन प्रोटीन असतो which is not a which is H1 histone is not a part of nucleosome. Nucleosome फक्त आणि फक्त which is made up of 8 octamer histone protein and wrapped DNA, that is the nucleosome. But histone protein which is the subsidiary role or extra protein, that is the H1 histone protein which is the subsidiary role or extra protein. And in that next concept is beads on a string, that means in Marathi manyanchi maal. Then what is the beads on a string here? Nucleosomes are repeating unit here of chromatin which are stained bodies present in the nucleus, and it is look like a beads on a string. म्हणजेच न्यूक्लियोसोम्स इज द रिपीटिंग युनिट. भरपूर न्यूक्लियोसोम्स, मण्यांची माळ जसे तशा प्रकारचे न्यूक्लियोसोम्स हे DNA ला काय झालेले असतात, बाइंडिंग करत करत जातात. दॅट इज द युनिट ऑफ क्रोमॅटिन विच आर स्टेन्ड बॉडीज विच आर कलर्ड बॉडीज प्रेझेंट इन द न्यूक्लियस, अँड इट लुक्स लाईक, आणि ते कशा प्रकारचे दिसतात, बेड्स ऑन अ स्ट्रिंग. हिअर 6 nucleosome get coiled to form solenoids. Solenoids which is the structure which have 6 nucleosomes get coiled. Solenoids means wiry like structure, like a telephone wire. अशा प्रकारचं जे स्ट्रक्चर आहे, that structure which is the solenoid. म्हणजे भरपूर न्यूक्लियोसोम्स हे एकत्र येऊन जशा प्रकारचं टेलिफोनचं वायरची जशी गुंडाळी होते तशा प्रकारची गुंडाळी, दॅट इज अ 6 न्यूक्लियोसोम गेट कॉइल्ड टू फॉर्म सोलेनॉइड स्ट्रक्चर अँड विच इज 30 नॅनोमीटर, दॅट इज द 300 एंस्ट्रॉनॉट एवढं त्याचं काय असतं लेंथ असतं. अशा प्रकारची हे सोलेनॉइड आहे. देन फर्दर सुपर कॉइलिंग विच टेंड्स टू फॉर्म अ लूप्ड स्ट्रक्चर which is called a chromatin fiber. First which is the nucleosome, first DNA, then nucleosome, then solenoid and the further structure which is the chromatin fiber. What is the chromatin fiber? That is the supercoiling which tends to forms a looped structure, that structure which is called a chromatin fiber. And then chromatin fiber, after chromatin fiber which further coils and condensed, आणखीन coiling होते, आणखीन condensation होतं. And at metaphagic stage we know that the metaphagic stage chromosomes are the advanced condensed state chromosomes here, and that chromosomes which is formed first DNA, then nucleosome, then solenoid, then chromatin fiber and then chromosome. The packaging of chromatin at higher level, this packaging of chromatin which is the higher level of packaging and that need the additional set of protein. We know here that the histone protein which is the that need the packaging, but the additional set of protein that are called non-histone chromosomal protein which also help, which is the additional set of protein and the packaging of the DNA, which is the packaging of DNA in the prokaryotic. Here we see the diagram of the nucleosome. Here the one nucleosome and that 8 octamer, here the 8 histone core we see here clearly, that is the green structure here, and red colored wrapped DNA we also see here, and that 8 histone core which is the nucleosome, and here standing structure, that is the H1 histone. Here the H1 histone is the additional proteins here, H1 histone here, that is the nucleosome. Like that the nucleosome, and many nucleosomes are formed. Here it's on a string structure here looking like here, that is the nucleosomes, and 1 nucleosome, 2 nucleosome, 3 nucleosome, 4 nucleosome, 5 nucleosome. Here the many nucleosomes, it look like beads on a string structure, मण्यांच्या माळीसारखं स्ट्रक्चर दिसतं. and 2 adjacent nucleosome, that is the DNA which is the linker DNA which is called. Then here the chromatin fiber or the solenoid structure here, that is the 30 nanometer here, the chromatin fiber structure we also clearly see here. Here the chromatin fiber, here we see that the structure, that is the beads on a string structure in the nucleus, that is then we see here the 11 nanometer beads here and the 30 nanometers chromatin fiber here. This type of the packaging is present in the eukaryotic cell. I hope you understand that. Thank you.